Okay,
So
perfect.
hello, yeah, hello everyone. Welcome to our phenomics webinar. It's my pleasure to start the salinity stress webinar. So it would be the first topical webinar that we will do. It's the second webinar after the summer break. I hope you could enjoy the summer break and now continue with the webinars, and we will have the salinity stress webinar presented by Jasper from the Wageningen University in Netherlands and Mariam Awlia from the University of Cape Town, South Africa. Some background about the organizers of the meeting and the goals of the webinars. So what we do, we set up a phenomics webinar as an exchange platform for the community to address a relevant topic in plant phenotyping. We are very interested in seeking for contribution from plant phenotyping related fields, basically anything that's related to plant phenotyping across all career stage levels and from research and industry. We do this two different formats. We have the single talks, so one large talk about a specific topic, and the thematic sessions where we have multiple talks, at least two, and focus on a specific topic like today. The webinars take place every second Friday, 2 o'clock, and so please watch out. The next webinar will be in 2 weeks. Some background on the organizers. So the webinar is organized by IPPN, the International Plant Phenotyping Network, which is an association which aims to integrate and advance plant phenotyping approaches. The goal is, or the mission is, to integrate the fragmented activities across the globe, facilitate exchange of knowledge, information, expertise among partners and beyond, address technology development, implementation, and advise also policymakers, funders, advise different stakeholders on future opportunities. There is a large number of stakeholders, people involved in that, and activities within IPPN, basically to bring you all together to facilitate a discussion. Additionally, EPPN 2020, it's a Horizon 2020 funded project, which facilitates access to 31 phenotyping facilities in Europe. The access is based on a simple application procedure. Once your access is approved, it's entirely for free. So basically all the costs are covered, including your travel costs to the facility. 20% of the funds are from non-European labs. So it's not only for Europe. And as you can see on the right up, there is quite a number of people who joined the EPPN 2020 call or applied from countries outside of Europe. And the calls are every 6 months. Unfortunately, we have right now the last and very final call, the 6th call. The deadline is in October 26th, so there is still time to apply, and hope to see you within the framework of EPPN 2020, which resulted so far in participation applications from 33 different countries and 124 ongoing projects, more than 70 publications. So I can just recommend you to use this opportunity. Additionally, the emphasis infrastructure project. So it's a European infrastructure project with a goal to enable a long-term perspective for service provision to the phenotyping community within the controlled conditions facilities, intensive fields with a high level of technology, high level of precision to measure different traits on the controlled, semi-controlled conditions in the field, network of field sites, but also data computational services and modeling are a large part of emphasis or portfolio of infrastructures around which services will be developed. The goal is to develop plant phenotypic infrastructure across Europe, provide access to this multi-scale phenotyping facilities, enable genotyping of performance, enable measurements of performance of diverse genotypes across different environments, share knowledge and novel technologies. Additionally, we have the webinar is broadcasted also in China via our partner platform, which is Agri Forum. So we usually have quite a number of visitors or from China. And last but not least, Jennifer Clark. She's providing the technical support for the Zoom platform right now, and we are very grateful, Jennifer, that you are doing that. Moving on to the speakers of today, so a very short bio. So we have Mariam Awlia from the KAUST. Actually, she did her master's and PhD at the KAUST University. She earned a scholarship by the KAUST University to obtain these degrees. She then moved to the University of Cape Town, is a postdoc there since 2 years. She's an expert in plant sciences, plant genetics, science communication, experience in big data analysis using R statistical programs R and then the second speaker is Jasper Lammers from the Wageningen University in Netherlands. He's an expert in plant sciences, image analysis, transcriptomics, R analysis, R programming, Python programming. He's a PhD candidate, I suppose, in the last stage of your PhD program. He did also his master's at the Wageningen University and was also active in UK and Australia. And as such, I do not want to waste or use your time for the presentations. So we would move on to the presentations now. And the first one would be Jasper Lammers from the Wageningen University of Netherlands, talking about how plants sense and respond to salinity. So Jasper, the stage is yours.
Sorry, I was muted. Thank you for the introduction, Roland. Indeed, I'm in the third year of my PhD in the group of Krista Testerink. And today I will talk about how plants sense and respond to salinity with a focus on the search for sodium specificity. So first I will start with a little introduction. Salinity is an elevated level of ions in the soil, and 7% of all land is affected worldwide, and this percentage is increasing. And I think even worse is that 20% of irrigated land is affected, and these are of course our most productive agriculture we have. And when we talk about salinity, it's mostly about sodium chloride, as this is the main source for increased soil salinity. And most crops are not salt tolerant, and that's why salinity is a major threat for food production. On the plant level, salinity induces two types of stress. One is a rapid stress, which is osmotic, and the ions in the soil, they decrease the plant water content. And the other one has a slower offset, and is the ionic stress, which the ions slowly accumulate in the plant, and there they inhibit cellular processes. And I think the most famous one is that photosynthesis itself is inhibited. However, like is the case with many abiotic stresses, we don't know much about the early signaling and the sensing of this. So this is also the case for salt stress. We know that intracellular levels of cyclic that the intracellular levels of calcium are quickly elevated, as are cyclic GMP levels and reactive oxygen species. However, we don't know the exact mechanism how sodium enters the plant cell, or how this is sensed, or which calcium channels are involved in this response. And this is also the main focus of my research, is to look at these early responses and try to dissect it. And of course, this is hard because we have this osmotic and ionic component happening at the same time. Oh, and what is interesting about the ionic component is that it has a slower, the stress is starting later in the plant life. However, we notice that it's already sensed in a very early stage. In our lab, we mostly look at roots, because this is of course the first tissue that comes in contact with the high salinity. And we see various responses, which I've listed here. And then on the, so And on the left, we have the control situation, and on the right, the sodium chloride situation. So one is the sodium avoidance, also called halotropism, in which we have introduced a gradient. And then the roots, they bend away from the sodium, as can be seen here. I will talk more about it later. And the other thing is that the root system is changing by sodium. So here on the control situation, we see a healthy root, but when the plant is treated with sodium, we see that the main root is shorter, the lateral roots there are less lateral roots, and they are also shorter. And then lately, we also see that the root morphology itself changes. So if we transfer 4-day-old plants to a plate with sodium, we see that the root morphology is changing because it's thickening here. While the other plant or the control plants, they have like this uniform density in the root. So we see this thickened thing here, but we don't know exactly what causes this, so I will not elaborate it during this webinar, but I will talk about the other two responses. So halotropism is the effect where the tropism where the roots avoid the high salt concentration. And how we test it in the lab is that we grow plants for 5 days on an agar plate without salt, and then we replace this here depicted in orange, the agar with a high sodium concentration. And because the roots are not directly on the border, they will not sense the salt immediately, but then there will be diffusion from this high concentration of sodium towards the medium, and then the plants sense from which the side sodium comes, and then they do bend. And what has been discovered is, I first have a video of this. I hope you can all see it. So here are 4-day-old plants, and they are growing first for 24 hours. And then the gradient is introduced here below, and then we immediately see that the roots, they start to bend away. And what's nice of this response is that we can quantify the response by measuring the angle. And that's seen here and also in the later picture. So if the root is growing down, we say it has like a direction of 0 degrees. But if there's an halotropic response, there's a negative angle. Well, if it goes here to the right, we have a positive angle. And then in the control situation, we see that there's a positive angle of roughly 7 degrees, meaning there's like a natural skewing a little bit to the right. But if we give the sodium treatments, we see that the root starts to bend away, as we also saw in the movie. And this is a concentration-dependent response. And what is very interesting is that if we have ionic controls like lithium chloride or potassium chloride, or osmotic controls like sorbitol, we don't see this bending happening. There's no significant change of root growth direction, meaning that this response is sodium specific. And that's of course very interesting if you are looking for a sodium sensor to have this sodium specific responses. And what was discovered is that this root bending is an auxin dependent response. So in a control situation, the root is growing down because there's an equal distribution of auxin between the left and the right side of the root. But when there's sodium detected, then the pin proteins, which are the auxin transported, they are removed from the plasma membrane, and less auxin is then distributed to this side of the root. And auxin is an inhibitor of the root cell elongation, so meaning that the cells on one side become longer than on the other side, and therefore we see this bending. However, not much is known about what initiates these responses. And on first sight, one could say that this looks a lot like hydrotropism, where the root bends towards the water. However, it was already shown that this was not auxin dependent. And in the lab of Malcolm Bennett, they've also seen that this was ABA driven. And they discovered that the cortex layer, which is a kind of tissue in the root, I will come back to it later, is the most important tissue for it. And in our lab, we don't see this response with 400 millimolars, as I showed before, but we do see it happening at much higher levels, like 800 millimolars of sorbitol. So these halotropism and hydrotropism are very distinct responses. And as I said, this was ABA driven, and we were very interested whether other hormones are also involved in halotropism. So I have a small overview of the ABA signaling here. So in the absence of ABA, PP2Cs, they inhibit ABA-dependent SNRKs, which are Arabidopsis has 3 of them. And then there's no ABA response. But in the presence of ABA, pills inhibit PP2Cs, and then SNRKs become activated. And this then leads to an ABA response. So if we have either, and we tested pill and SNRK mutants to see if ABA was important for halotropism. And indeed, this was the case. So this has a bit of different value on the y-axis, and so I made an overlay of the control root, which grows like this, and the sodium root, treated root, that grows like this. And here the angle means the difference between the sodium roots compared to the average of the control roots. And we see indeed that the ABA mutants have much less response to the sodium. And then we are, because this is quite striking, because hydrotropism is a different response, but we also see this ABA dependent. So we were also interested whether the same tissues were involved in this. So again, here we have the Columbia and then the double mutant, which bends less. And then if we express SNRK 2.2 under its native promoter in this SNRK 2.2, 2.3 background, then we can see that we restore the response. We received these lines from Malcolm Bennett. So these were the same as were used for the hydrotropism study. And then the roots, they have the root has various tissues, starting with the outermost layers, the epidermis, and then followed by and this is also where the pins 2 are located. And then the layer next is the cortex, followed by the endodermis, and in the middle is then the stele with the vascular tissue. And what we saw that if we express SNRK 2.2 with a tissue-specific promoter in this SNRK 2.2, 2.3 background, we see that the cortex, the root cap, or the meristem, if it's expressed there, it's not able to fully restore the response for halotropism. But if it's expressed in the endodermis or in the epidermis, we do see a full restoration of the response. So this shows that different tissues are important, and of course this could also maybe help us to find the sodium sensor and in which cell layers this is happening. But yeah, we don't know much more now. We are further investigating this. Then the second part of my talk is about the root growth dynamics. And in the group of José Dineni, they showed that if roots are salt stressed, that the roots, they quickly decrease growth. And then they enter a state that's called the quiescence phase, in which there's very little growth, after which the growth is restored partially, but not complete to the original level. And here it's already shown that different tissues can have different growth rates, which could be important for the different strategies we see of Arabidopsis. So various ecotypes of Arabidopsis, or the ecotypes of Arabidopsis have 4 different survival mechanisms. So either investing in a main root, the lateral root length, or lateral root density. And of course, by varying this recovery rate, we could maybe end up with this could be the cause for the different strategies we see. And these growth dynamics, I mean, this is happening in the first few hours, so we really need time series of growth to see this. And for this, we have a time lapse set up in our lab, in which every plate is imaged once every 20 minutes. And here's a fast forwarded video of it. So we see the camera moving along all the plates. And in the back, there's also an LED panel moving with infrared light. So this also allows us to image the roots in the dark. So 24 hours. And then the images look like this. So we can clearly see the roots and follow the growth. And then interestingly, if we look at the responses, for example, for sodium chloride, we indeed see, like was reported, that there's a rapid growth, decrease of growth. The beginning of the decrease is not shown here because of handling and the time it takes to start the machine. But then we do see this quiescence phase, after which the growth is recovered, let's say after 5 hours. but we do see the same response for potassium chloride. So it shows the same trend. And interestingly, this is not happening with sorbitol. So there's this decrease of growth, but then the growth is quickly recovered. So it doesn't have this quiescent phase. And this data shows us that there's that this is an ionic response. So not sodium specific, but at least happening with potassium chloride and sodium chloride. So with halotropism and this study of the quiescent phase, we can maybe already move towards a sodium center by having by making a clear distinction between the phenotypes we see. But then we know quite some mutants that have effects in the roots under salinity. However, we know very little about the shoot and the shoot growth dynamics. So we wrote a proposal, and that was granted by EPPN to work with the Phenopsis system in Montpellier, INRA Montpellier. We worked with Bertrand Muller and Mariam Dozat. And this machine this platform has very precise control of all the environmental variability, environmental factors, and therefore biological variance is limited between the replicates. And this allows for a very very robust data that we can use to study this growth dynamics. And this system also has a camera that can make pictures every 30 minutes. So also here we can look at this, these quick growth responses when salinity is added. And then the plate from above looks like this. And also there's an infrared camera. So we can also, this allows also for imaging during the day and during the night. What we did is that we injected plants of 22 days with sodium treatment directly between the roots to have this very quick response. So we at least know that the quiescent phase is happening, but we're also very interested in the shoot dynamics then. And then by imaging, so this is the control situation, and the relative growth rate is plotted. So we see there's this distinct day and night rhythm with the night when the night in the gray, and then the day of when the lights are on in the white bars. So there's this distinct mechanism. I'm just showing here the SNRK 2.2 as an 2.3 as an example, but we tested various mutants. But then when we add sodium chloride, indicated here by the red bar, we see that this day-night rhythm is different after. And also there might be a quiescence phase, but we don't, there might be a quiescence phase. However, because we use the camera from the top, we do not really know whether this is only growth or that also maybe leaf movement is affected, which of course changes your projected area. So more research is required, but at least we see very interesting patterns already. And then lastly, we see, of course, all these root growth responses. We are also very interested in what's the transcriptional basis for this. So we did an RNA sequencing on 8-day-old roots. We treated with sodium chloride, sodium nitrate to get the sodium, to get the effect of to distinct the effect of sodium. And we also had a potassium chloride and a sorbitol treatment for the ionic and the osmotic treatment. Stress. And then we harvested roots at 6 and 24 hours after we did RNA isolation and sequencing, and then the analysis with DSEC2. And after we did clustering of the genes and treatments, geoterm enrichment and motif enrichment to find promoters. And what we saw is that, so here are the most affected genes on the Y or on the X-axis, and on the Y-axis are then the treatments. And what we so we get the different clusters here, and we see that many clusters, they are not very different between the treatments. So here we get in blue is then the downregulated, and in red the upregulated genes. So we see that, for example, the first cluster has a lot of downregulated genes, but there's not so much different between the treatments. And the same holds true for the other clusters, more or less. So here we have a very highly upregulated, here a bit less, and then a bit less again. But I mean, all the clusters are very much the same. So I would say this could be the osmotic response. But the last cluster shows a clear distinction between the sodium treatments, the lower two, and the other treatments for most genes. So there's also a sodium-specific response going on here. And by using these genes, we could do a GO term enrichment, and then we found various GO terms, of which this is a summary. So we see quite some responses to biotic stress, cold and hypoxia responses, but there's also many transcription factors in this cluster, possibly required for further downstream responses. And then we could predict which transcription factors are important for these genes by looking at the motifs in the promoter region. And we find also many transcription factors where they all belong to the same family. So one is the NEC family proteins, which are known from drought and cold stress. We find quite some workies which are also activated by various stresses, and also quite some ABA-related transcription factors. And currently, we are investigating mutants to see of these transcription factors to see if our prediction is right and to see whether they are important for sodium stress. And then I already come to the conclusions of this talk. So halotropism is mediated by a tissue-specific ABA regulation, which is very different than hydrotropism, and it is sodium-specific. Then the root quiescent phase is an ionic response, which is then, which opens new questions about the regulation. And then also we see that many genes in the transcriptome, they are regulated by, I would say, an osmotic component. Well, a small fraction of these genes are regulated by sodium specifically. And this research was made possible by many people, which Christa Testerink and Charlotte Gommers, they are supervising this project. IODGC did a lot of work on investigating the halotropic response. Eva van Zelm and Jessica, they are working on the quiescent phase. And Jessica and Yangsha, they help with the transcriptomics. Then Bertrand Muller and Mariam Dozat, they worked with they helped with the Phenopsis. They are really the experts on Phenopsis. And then Malcolm Bennett for providing the ABA tissue-specific lines. And thank you for listening.
Are there any questions? Or actually, what I missed to tell you, you can at any time during the talk, you can write your questions into the chat. There is right now one question, but feel free to write any other questions, and we will pass the questions to the speaker. So there is right now, Jasper, there is a question, KCl and NaCl response. Is it sodium or chloride sensor?
You mean in both of them, or you mean the ionic response?
Yeah.
Yeah, one could say that maybe so we don't know much about the sensors of sodium or potassium chloride, so it could as well be indeed an ionic sensor, but we don't know yet. Does this answer the question?
If it does not, then it was a question by Ron Sellingman, so please specify some more details if you want to know more. So there is another question, did you do any tissue-specific study on auxin-related responses, and how does tissue-specific response relate to auxin?
No, I haven't investigated myself, but no.
Mm-hmm. Any idea about auxin response? Auxin, what role plays auxin in this entire and this tissue the differentiations from literature or any other resources?
Yeah, so the PIN2 proteins, they are active in the epidermis. So one could say that for sure the epidermis is required for the auxin responses, but also lower in the root. There are also other PIN proteins. So yeah, they could of course also be important for the distribution of the auxin. Yeah.
Okay. Any other questions? Then I would suggest that we move on to the second talk. There will be, as already mentioned, so please feel free to add the questions during the talk into the chat, and we will have after we can respond to the questions in the chat, and in the end, both speakers can also respond to questions if there are any general questions. So Jasper, as such, thanks a lot, and we can move on now to Mariam, and the stage is yours.
Hello, does everybody hear me? Okay.
Yes.
Thank you for hosting this very interesting webinar, and Jasper actually already paved the way for me, so there's a lot of overlap, and you will see that I will expand on things that he actually just mentioned. So my name is Mariam Awlia, and I did my PhD in genetic mapping of the early responses to salt stress in Arabidopsis thaliana. And my PhD took place at King Abdullah University of Science and Technology, which is in Saudi Arabia. And this was led by Professor Mark Tester, who is somewhere in the audience. So in order to put into context how salinity is a worldwide problem, this global map just shows you how severe and widespread this level of toxic salinity has reached the global scale. And there's been an estimate from 2014, but I'm sure it is aggravated now, of an estimated loss of $30 billion a year due to this soil problem, which causes crop reductions. And here is Saudi Arabia, and you can see that it's quite salt affected, which affected my motivation to work on this research. But let me put into context what happens to plants under salt stress. So if you can see these two plants, and we can compare their relative growth rate through time, under controlled conditions, it's relatively stable. But when salt stress takes place, there's a rapid reduction in growth. And Jasper also mentioned this. The osmotic phase is, at the moment, not very well understood. But in our case, we labeled it as shoot ion independent. And I will arrive to that point again, just to emphasize why we say this. This response seems to take place between hours to days. And the first or the most prominent symptom is a reduction in growth. But with time, as the ions reach a toxic accumulation in the shoot, which we term the ionic phase, there is a symptom of premature senescence. If you can look at this prematurely senescent leaf becoming more yellow. So the way we distinguish these two phases is by saying that this is shoot ion independent, and this is the result of ion accumulation. Now, if we put this on a time scale, of course, this is relative and hypothetical, but we hypothesize that there's a rapid signaling that takes place and a change in hormone accumulation, including abscisic acid, osmotic acid, and all of that. But these processes eventually cause a decrease in transpiration and reduced photosynthesis, which ultimately affects growth. And then we hypothesize that ion accumulation takes place and causes other damage, leading to premature leaf senescence. So basically this square here is relatively unknown and understudied, but was the topic of my PhD. So I applied forward genetics in my work to identify the genes that are potentially responsible or contributing to the early responses to salt stress. And in my work, we developed protocols for measuring these early responses. We also phenotyped mapping populations to genetically map loci using genome-wide association studies, and to finally identify candidate genes that hypothetically could contribute to these responses. And since we are talking about phenotyping, the machine that I used is called Plant Screen, and it has red, green, blue imaging platform, RGB for short, and chlorophyll fluorescence. And this was sponsored by Photon Systems Instruments. So for the RGB imaging, this is a snapshot of how the process takes place. We take a normal RGB image. It's corrected for this fisheye image, and the background is subtracted and then processed to give information regarding the morphology, which you see here on the left, related to the pixel counts, or looking into the colors and how that changes through time, which was quite interesting in my work, because I don't think it has been studied in depth in terms of stress. And as for chlorophyll fluorescence, I just wanted to give you a little explanation of what happens to plants when they harvest light. So here you see light being harvested by the leaf, and then you have mainly three reactions: fluorescence, photosynthesis, and heat. And these are three antagonistic reactions. There's a little bit of reflection off the leaf, but that's negligible in our case. So when a plant is under stress, there is less light harvested for photosynthesis, and more light is, or photonic energy, electron, whatever you can understand from that point of view, it's re-emitted as fluorescence or released into heat. And you can see here on the right, different equations or parameters that measure these components, as you can see here. And in terms of the phenotyping, this kind of graph would be generated as a result of chlorophyll fluorescence imaging. And you can see different traits are highlighted based on which point the camera took a measurement of the fluorescence intensity with different light bursts, as you can see here. For example, if we take the first box on the left, we have the minimum fluorescence, which takes place without any excitation, and the maximum fluorescence that the plant releases or emits. And if these pictures are compiled into frames, you can see the image here on the right showcasing different levels of intensity, which are calculated to provide parameters reflecting different parts of the electron transport chain. And to put into perspective why we use mapping populations, they are very useful in forward genetics to identify causal genes. And in my case, I used the Arabidopsis HapMap population that has a wide range of accessions, very high density genotypic data. The one I used, the second part of my PhD, was this 10 million SNP marker dataset, which is super useful. And it has a lot of mapping tools available online. And here's an example of a study that successfully used GWAS to identify this very famous iconic gene that is called HKT1, high potassium transporter 1. And you can see that it's very well defined in this peak. This was a study conducted on the natural variation across coastal plants or accessions of Arabidopsis, but it is related to shoot ion accumulation. And as you can see, we are trying to study the segment of these responses that are shoot ion independent. So in terms of my protocol, I planted the Arabidopsis accessions, and at 2 weeks, I controlled the watering, which is something that we developed in a previous publication. And so we watered it to 60% of the water holding capacity to control how much salt is in the soil. And at 3 weeks old, 10 to 10 leaf growth stage, we implemented 250 millimolar sodium chloride as a treatment. And this resulted in a final concentration of 10 millimolar sodium chloride. And then we immediately performed the phenotyping. Within 1 hour, we performed chlorophyll fluorescence imaging, the RGB, and we took weight and watering measurements. And these are some examples of the results. These were the plants that I showed earlier. You can see how quick the change took place. Within a week or even 4 days, you see the difference between control and salt stress with a reduced level of growth and shoot expansion. Here's an example of a change in greenness, which I will discuss more in depth later on. And then these are examples of the change in chlorophyll fluorescence. So in terms of the change in growth and change of area through time, as you can see here across 191 accessions, we have a box plot showing the control plants in blue and the salt treated plants in red. And immediately you can notice from day 2, there's a significant difference between these two groups of plants by condition. And if we take one plant, for example, Colombia, you can see how at day 4, we found a significant difference between the conditions. But I also wanted to highlight that we wanted to separate the two intervals to reflect the early and late salt stress responses, hopefully relating to the osmotic and ionic phases of salt stress. And you can even see from the salt stress linear fitted growth rate, it's relatively linear, while from the control perspective, you can see that there's a slope change between the two intervals. Of course, this is dependent on different accessions, but we wanted to use it as a proxy, and we created this ratio where we would take the growth rate in salt divided by a growth rate in control in the two intervals. And this was quite interesting for us to use, because when we plotted these two ratios of the shoot ion dependent tolerance index, or salt tolerance index for short, across the accessions, we see that there is no specific correlation, indicating perhaps that these two phases are actually independent and they have unique genetic constituents, which was very interesting for us and exciting to see what genes will come up with the GWAS. In terms of the change in color, you can see here that between control and salt stress, there were a wide range of changes and a lot of significant differences. However, we couldn't attribute these changes only to salt stress, because these are also natural changes due to plant development, which need to be further characterized and studied. In terms of the chlorophyll fluorescence results, here are just two examples of minimum fluorescence measured in the light adapted state. You see here with the prime symbol, and minimum maximum. Unfortunately, we lost the data from the first 2 days, so I apologize, just to show you from day 2, but we can see that already significantly different between the salt and the control treatment groups, which is quite exciting and very rapid. Here I would like to just give you a snippet of the types of data traits that we added to the GWAS and the number of traits that we were able to incorporate. So here you have RGB traits on the left, and the green is hues, chlorophyll fluorescence, and the traits that are colored highlight traits that have significantly associated SNPs from the GWAS. But just to highlight here, we measured these traits across 8 days with the inclusion of relative growth rate measurements of interval 1 and 2 across 2 treatments, and with the shoot ion-independent tolerance index of interval 1 and 2, giving us a total of 438 traits, which is fascinating for us to have that much data to dig in and look at the changes across traits, which unfortunately I don't have time to show you now, but we are working on producing a publication soon. In terms of the GWAS results, this was the most exciting. So we have single and multi-trait GWAS models, which was provided, the multi-trait was provided by Arthur Korte. And in this case, you have a GWAS that was independently run with the control measurements. And this was quantum yield that was measured in the light adapted state on day 3. And the same trait measured under salt stress. And here's a nice peak that came up on chromosome 1. The added bonus of adding the multi-trait model helped us perform this interaction where we could test whether this effect between the two traits is genotypic, or is it environmental, or is the association, affected by genotype, environment, or the genotype by environment effect. And we can see here that there was no association found in the genotype effect, which emphasizes that this association was salt stress specific. And this is good news for us. We saw the same SNP in the environmental effect test, as well as the G/E test. So now if we zoom in to the locus, there were about 50 SNPs that covered, so that we said the locus would be around 10 kb, because this is the linkage disequilibrium, the average linkage disequilibrium in Arabidopsis. 10 upstream and 10 downstream, so we have around 20. We focused on some of these genes during my PhD, but I didn't have enough time to identify all the candidates, but it is part of the new publication that we're working on now. Interestingly though, the locus is largely uncharacterized, so it's green fields. We are able to test these genes and see how they're involved in salt stress or other processes. Now, to summarize, this study was able to put the different changes on a timeline, at least hypothetically. What I saw first was the change in rosette coloring, which was as rapid as 1 hour after salt stress, followed by a change in heat dissipation or photosynthesis, photosynthetic rate, and the photochemistry. This later caused a change in rosette area and a change in rosette shape, as we say here, architecture, but including like compactness and slenderness. And then finally, we saw a decrease in the quantum yield, which is relatively the most robust parameter in chlorophyll fluorescence imaging, because the plant tries to retain this ratio for its survival. And in summary, we were able to develop a high-throughput phenotyping protocol for other people to use and apply in other types of stresses as well. But in our case, we looked at salt stress. We demonstrated that early and late responses to salinity are not correlated. We captured the natural variation across almost 200 accessions. And then we examined trait relationships. We applied multivariate analyses, which I didn't have a chance to explain here, but hopefully you'll find more details in our publication. And we applied both single and multi-trait GWAS to identify salt stress specific associations. And we detected over 750 unique loci that were significantly associated with salt stress. And we also tried to filter out these associations to see which ones were repeatedly being highlighted across different traits. just to add more weight to these candidate genes, that they affect more than one trait and could be more useful for future breeding. And we phenotyped homozygous candidate gene mutants, but again, we needed to perform more outside of my PhD, which we have done now in the past 2 years. And when I was still performing this research, we looked at least these 2 genes and hinted that they are involved in early stress responses of salinity. And in terms of future work, there's always room for more phenotyping, especially gene mutants, and finding more candidate genes that are homologous to the ones in Arabidopsis in crops, because ultimately we are aiming to provide more breeding targets for improved salinity tolerance. Other types of traits that are interesting could be measuring transpiration rate and adding the component of water use efficiency, or using thermal imaging just to look at temperature changes, exploring the correlation between photosynthesis and transpiration, also including hyperspectral imaging, which has huge potential in data mining and understanding plants, and finally examining the ion accumulation while comparing that to the early responses to salt stress. And with that, I would like to deeply thank the team at KAUS, the Salt Lab, so Magda, Mark, Sonia, Nov, and Marshall, who helped me a lot at the end of the PhD, and the team at PSI, Clara, Jiří, and Martin and Radka. They were very helpful in helping me develop the phenotyping protocol. And we have Arthur, who supplied us with the multi-trait mixed model GWAS, Xavier, who helped with setting up the optimized watering and salt treatment protocol, and Stuart Roy, who thought of this concept very early on before I even came on board in the Salt Lab. And with that, I thank you for listening, and I welcome any questions.
So thanks a lot, Mariam. That was a really nice presentation. I think there will be some questions. Yes. So the first questions by Darmodar Poidal. So plants under heat stress, early vegetative and generative stage response found correlated. Just a second, needs to get the question right. So the question is, plants under heat stress, early vegetative and generative stage responses found correlated and also reported validated under field conditions. What could be the reason in case of salinity in the stage response where if the interstage response were not correlated? Sorry, it's a little difficult to...
Well, the easy answer is we don't know yet. But I don't understand. The beginning of the question said heat. Is it heat stress?
It was also heat stress, yeah.
I'm not sure. The thing is, I'm not referring to heat stress in the plants, but that the plants release energy in the form of heat. Just like humans, when they are stressed, they would sweat. So the point of measuring heat dissipation is just that it's another proxy to see if photosynthesis has decreased or increased. If photosynthesis increases, heat dissipation decreases. And of course, fluorescence also, they're the 3 antagonistic reactions because the photonic energy is harvested or let's say absorbed, and it goes through these 3 channels. So I don't know how that's relevant to the question. Heat stress has a different process in the plant, and I have a colleague, Noah, who actually looked into that, but I don't know how this can be relevant to salt stress exactly in this context.
Mm-hmm. There was also this aspect of early vegetative and generative stages. Would you expect any differences in response between those early stages and later stages?
Absolutely. Like I said, we've only just begun understanding the mechanistic physiological changes, but surely there must be different genes that are activated in these two stages, especially when we separated the intervals and performed GWAS on these different time points or phases of salt stress responses, and they seem to have different genes. Perhaps we can even say, in terms of common sense, perhaps seedlings are more susceptible than larger, bigger plants, just because plants are able to allocate their nutrients and energy to different components. I actually have another slide that talks about the three types of tolerances or mechanisms of tolerance that Munz and Tester hypothesized in 2008. One is the osmotic component, which relates to signaling and changes in cell activity. The second is ionic transport. So they would sequester, or not sequester, but move the ions away from the shoot, just to reduce the risk of affecting the photosynthetically sensitive parts of the plant. And then finally, tissue tolerance, where the plant sequesters ions into old leaves or in the vacuole. So it's almost sacrificing parts of itself to die so that the rest of the plant that's younger can live longer. So I suppose that could be an answer to the question that, of course, younger seedlings have less leaves to perform tissue tolerance, and perhaps it is more difficult to sustain itself. Rather, in the case of a bigger plant, it's easier to have higher tolerance.
Mm-hmm. I hope, Darmodar, your question was addressed. So we would be interested in the response of photosynthesis, fluorescence to salinity. So probably the one of the response reasons is so it's not a direct response to salinity, but simply stomata close and photosynthesis changes. You mentioned right now also the salt transport. Potassium chloride, is there also a direct effect of ions, osmotic effect on photosynthesis that could also induce some reduction of photosynthesis?
Yes, absolutely. Again, we are not sure exactly how this takes place, but in terms of the ionic direct effect on photosynthesis, from what I understand, the ions could compete in the reaction centers of very important enzymes during photosynthesis. Perhaps it changes the not the osmolarity, but the electromagnetic gradients. Ions, when they are on the high end or low end in the cell, can wreak havoc because it's just not sustainable. Whereas indirectly, of course, with the reduction in growth, we are hypothesizing that it's not related to the ions. So it could be related to water. Like I was going to say, the osmolarity, it could be related to, yes, changes in stress hormones, closing the stomata, and changing other kinds of processes in the cell that we still don't understand, cell cycle activity. So, of course, I think there can be direct and indirect effects, but there's still a lot more to uncover.
Mm-hmm. Yeah, thanks. There is also a question, if you studied on salt-induced drought in the plant during your study, so basically the differentiation between salinity stress, drought stress, potentially combination of both. What would be your opinion? What do you think about that?
Yes, great question. Just because I can put into context where I am now. So I'm now working on a group of plants called resurrection plants, and they are naturally able to go into this dormant state when under drought. They completely desiccate. And as soon as you water them, they rehydrate fully again. And my plan, well, I started actually already when I arrived for my postdoc, was to test whether these plants are also salt tolerant. They're already drought tolerant, but can they also sustain themselves under salinity with combined drought and salt stress? And interestingly, we only did a few pilot studies so far, but one that is relevant to this question. So we had 12 plants, and we added salt stress to pairs just to have replicates of each treatment, because we wanted to just identify which salt stress concentration we should use next. And we went up to 500 millimolar sodium chloride, which is a lot. And I wanted to just test how far these plants can tolerate salinity. So we performed the salt stress and left them to dry out. And I remember we left the plants for at least a month. And then we rehydrated them. And only one plant regenerated and rehydrated itself. I think it was 200 millimolar sodium chloride, if I'm not mistaken. We did 600, 500, 400, 300, 200, and 100. I think, and there must be other smaller treatments that we tried, but this was the most interesting. And thankfully, we are going back into the lab soon. And the PhD student that I'm working with, who is responsible for this project, will look more into how we can differentiate the genes that are related to drought or salinity. Can we have more tolerance to salt stress with drought tolerance too? And the answer is, I think, yes. This is the long version of the answer, yes. Hope that answers your question.
Yep, thanks a lot. There is a methodological question. Could you elaborate how you recorded associated data? So basically image analysis, what tools did you use? I think it would be a question about the phenotyping platform, the data acquisition, image analysis.
Yes, so I performed the first experiment in Australia in the High Performance Plant Center. I think that's the name, by CSIRO. And their machine was manufactured in Czech Republic. And then eventually I went to Czech Republic and performed the experiment there, which was PSI, the group that I mentioned here on the right. So if this person is interested in phenotyping at that facility, they're happy to support you. But also with the European Plant Phenomics Network, I applied for the grant to perform phenotyping with the resurrection plants at Wageningen. And interestingly, it was supposed to happen before lockdown, and somehow the email got lost. But now we're going to perform that soon. So the image acquisition takes place in this phenotyping setup. Maybe I'll show you a picture again. So automatically the plants are placed in these trays, and you have the imaging stations in the phenotyping instrument. Basically you press a button and then this goes around, and you already preloaded the protocol of how many seconds it will take to take this image. In terms of chlorophyll fluorescence, there are different protocols. Do you want to focus on quenching or something else that's called light curve, where you look at different photon irradiances and almost stress the plant at different levels with the same trait, measuring that same trait. So I suppose in order for you to understand the phenotyping itself, it's easier to look at the websites and the machinery of how that was built, because it was all automated. And this was provided by PSI to support my work, but there are other phenotyping high-throughput phenotyping setups, just like the one in Australia that Mark developed in Adelaide, which is called the Plant Accelerator. They also have a similar setup, but also designed by a different company. It's just the idea that you need to capture images or measurements repeatedly in the most automated way to reduce human error.
Thanks a lot. What I actually missed in this question, the first part of the question referred specifically to leaf color changes, change patterns.
I see.
Sorry,
No
any comments on the leaf color changes? Because I suppose that depends also when you do the same measurements in different facilities, then the background light and you name it, you need to take a lot of aspects into consideration.
Yes, actually.
Yes. Please go ahead. Sorry.
Yes, actually, it doesn't matter what the background or the lighting is, because it's a controlled environment in this phenotyping setup, and it's dark, so the picture is always taken with the same amount of light. And in order to explain to you the processing, I'll just say that we took several images from the pool of all the RGB images that we have. For example, if you look at actually this one, the one that's pre-processed and without the background, and we put it through a Python model to color segment it. We first identified 9 hues. This is why I have these on the right here. The 9 hues that were most represented in this pool of pictures. And then we based all the other RGB images on how many pixels they had of each of these hues. So the processing is actually the same for any picture that will be taken. So when we perform the same setup at KAUST, because we have the same plant screen instrument that was purchased from PSI, it still has the same processing. It's just that you take these images, identify the most predominant hues, and then analyze all the other images based on that. And then you can see if there was a change relative to that template analysis. Does that answer the question?
Yeah, I think yes. So it means in essence the data from Adelaide, KAUST, or Czech Republic are comparable in the end, which is the ideal case. Yeah.
Yes, I suppose it's best to compare within the pool of images. And I think if you highlight which hues exactly have been identified, then it's easier to compare. But I also feel it's difficult to make conclusions just because different plants have different plant development stages, and these hues could be related to many things, pigmentation or even cellular proximity. There's so many things we still don't understand.
There was also a question from actually it was a question, I suppose, related to Jasper, but you can maybe both respond to that. It was about the role of gibberellic acid in root growth and development, and especially when we talk about stress physiology. So gibberellic acid and root development, root growth and development.
I would suggest Jasper to answer that. That's okay.
Yeah.
These different phases that are described, like the quiescence phase and the recovery phase, there's quite some work done on hormones on a paper in the group of José Dineni. They really describe per phase which hormones are important. And they also looked at jasmonic acid, gibberellic acid, ABA, and breast steroids. So we haven't looked at it ourselves yet. But I think, most information is there, but I cannot recall the exact details as well.
Okay.
Yeah. Oh yeah, GA is mostly important for the stop of growth in the beginning. Mm-hmm. Yeah.
Okay. Thanks a lot. Any other questions from the audience? I think there are no further questions. So I would like to thank you all for the presentations. Let me just put just a second. Just to share the screen once again, so you thanks to the speakers, it was really great. It was a great overview about the salt stress phenotyping and your research, basically very relevant for the audience and for experts, and not only, I suppose. What I would like to share with you is also short information about the involved platform. So IPPN, emphasis EPPN. You find all those information about plant phenotyping you may look for on those different platforms. What we specifically would like to ask you also to subscribe on the newsletter for emphasis. So because of the new GDPR rules, we had to change the subscription, change the platform. So we would really appreciate if you could renew your subscription. And obviously also please feel free to apply for access, and watch out for look out for on the websites. There will be an announcement for the upcoming next webinar in 2 weeks. It's in the process of being set up. So within the next couple of days, we will announce that. So please watch out, subscribe for the newsletters, and we are looking forward to see you in 2 weeks. And once again, Mariam, Jasper, thanks a lot for your presentations. It's highly appreciated. We really enjoyed that, and it's a great way of closing the week. So thanks a lot, and have a good weekend, and see you in the latest in 2 weeks. Thanks a lot. Bye-bye.